Da, 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 da. Oh, hello. I suppose you've come here to learn about eukaryotic gene regulation and epigenetics. Well, you've come to the right place. But before we get into it, let's meet a few special guests and get a word from our sponsors. First off, this is brought to you by my home on a snow day, as seen by the lounge attire, the bags under the eyes, and the hot cup of coffee brought to you by the Washington Coffee Shop, which fuels me every single day. This energy doesn't come from just nowhere, people. Come on. Our two special guests who will only be joining us for a little bit are Pepper the cat. One moment, please. Say hello, Pepper. This is Pepper. She says hi. And Jack the dog. One moment again. Here's Jack. Can't you see him? He's so cute. Unfortunately, they can't join us for the whole episode today, but I did want to introduce you to them because, hell, they were a part of this process. Now, let's get into it. First, let me move my face for you. All right. We're going to address some big questions today. We're going to get to prokaryotic gene regulation when we come back from school, whenever the heck that is. But until then, let's check this out. I can't make it my whole screen, by the way, so we're just going to have the slides like this, but I think you can see them pretty well. All right, so we're going to look at how genes are turned on and off in eukaryotes and how cells with the same genes differentiate to become completely different specialized functions. So basically, we're going to look at how genes are turned off and on in specialized cells, because we know that they don't all express all their DNA, and they change which genes are expressed at different times. So we're going to look at how they do that, the mechanisms that they do that. And keep in mind, more and more information is coming out about how organisms do these things, and especially eukaryotes, because we're very complex multicellular organisms. So more and more information keeps coming out, and there's more and more investigations being done to learn more about these complex endeavors. Okay, so let's keep in mind that the goal of a eukaryote is to maintain internal conditions and a constant internal conditions in the cells, in your organs, etc. But we're going to focus on the cells today, while facing changes in the environment, changes in temperature, changes in nutrient availability, changes in you name it. There's all kinds of environmental changes. So basically, this is maintaining homeostasis. And this is how we're going to regulate our body functions as a whole, both in growth and development, when you grow from fetus to adult phase, and then also how your cells specialize, turning those off and on. So we talked about prokaryotes and how they regulate mainly at the transcriptional level, okay? Eukaryotes are more complex, and we know that because they have those organelles, especially the nucleus, which is the big organelle that differentiates prokaryotes from eukaryotes. And we know that they have multiple levels of control because of this, not just transcription in eukaryotes. So we're going to talk about these in order. Number one, packing and unpacking DNA. Transcription, the mRNA processing, how mRNA is transported, how translation occurs, and then you can even have regulation after translation occurs and you have your proteins. So at all of these levels you see here, we're going to have some level of control. So first is DNA packing. Remember that we talked about how your DNA is wrapped around these histone proteins. So you can see that in the diagram here. And we call those, when you have the histone proteins in the DNA actually wrapping around it, we call those nucleosomes. So that's what the nucleosomes mean here. And it kind of just looks like these beads on a string under the microscope. So that's what it looks like. Then if you take those histones that DNA is wrapped around, and then you coil it even more, just like you're twisting a shoestring over and over and over again, you get this complex, you get this really compact structure, which eventually becomes your chromosome. Okay, we know that already. We know that you can twist it, compact it, and we know that these histones help you do that. Okay, and some of those are called just loop domains, just when you get it twisted enough. Don't worry so much about that. So we focus on these nucleosomes, and this is where the first level of DNA unpacking is. And don't worry about the positively charged amino acids. Yes, they bind tightly to negatively charged DNA, and that's how they wrap well. But what really matters is how tightly they're wrapped around, okay? So the more tightly that fragment of DNA is wrapped around that histone, the less those enzymes can initiate transcription, and less those transcription factors can get there. And so the more likely that gene is to be turned off, okay? And you can see, I'm going to focus on this microscope image right here. We have two new words we haven't learned before. Look at this picture first and look at the darker DNA. And then there's some sections that are lighter in DNA, okay? So this whole section is DNA, nucleus full of DNA, and there are darker sections and lighter sections, okay? The darker sections are tightly packed. So that means that they're twisted, really, really condensed, and that's why they appear darker. Think of like when you're shading in art class. The more you shade, the more you shade, the more compact your lines are together, the darker it's going to appear. And the less lines you have, the less they're compact and the lighter it's going to appear. And that's the same here. So your euchromatin is your lighter DNA, and that's your more loosely packed. So that's going to be the lighter areas of DNA. The heterochromatin, or the darker DNA, actually is more tightly packed, so you're much less likely to express those genes in those areas. So you can control which genes are expressed simply by having some of those areas be more tightly packed than others. So if you're a specialized cell, like a white blood cell, and you don't necessarily need to make all those genes, the genes that you don't need to express can be more tightly coiled, and so therefore they won't be expressed. Those enzymes for transcription, the proteins for transcription, like transcription factors, won't be able to get to that DNA, and therefore it's not going to express that DNA. And you're not going to get mRNA, which means you're not going to get the proteins. So that's one way that that regulation can occur. All right, and we're going to look at this. I put this link on your Google Classroom, and it's Lick Some Rats, okay? I'm going to go to this here. Okay, so you can read this yourself just to go through. But this is one of the levels that can be controlled by your environment. So how tightly coiled certain genes are can actually be affected by your environment in early development. So they've shown this in rats specifically, okay, and you can do this yourself. We're just going to hit go. I might need to refresh that real quick for us. Okay, so it's loading. This is what you're going to see. Okay, so high nurtured rats and low nurtured rats. What that means is high nurtured means they're very well groomed and taken care of as youth, as babies, basically. And then low nurtured rats are generally ignored by their mother. Okay, so we're gonna hit go here.
In the brain of a newborn rat pup, methyl molecules, green, silence the GR gene. When it's active, the GR gene produces a protein that helps the body relax after a stressful event. The type of care a pup receives from its mother during the first week of life can change the expression of this gene.
Okay, so what you have here is you have one of the genes that makes a protein for the stress response, okay? When it's active, this gene produces a protein that helps the body relax after a stressful event. okay? The more tightly coiled up this region is here, the less likely you're going to be able to make that relaxation gene, okay? And this can be controlled in early life. So first, let's be a really good rat mom, and we're going to lick our babies like we should as good rat parents. So let's lick these rat pups. So look, ooh, I'm the mommy. Okay, we're gonna groom the rat pups. Ooh, 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 ooh. I'm very attentive parent. Look at me.
Wow, you're an attentive rat mother.
Wow.
It's surprising your pup has any fur left on its body. Your licking and grooming has seriously activated your pup's GR gene. Your pup will have an easy time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So that's kind of weird, right? And that occurs in, there's studies that show this occurs in humans too, based on the trauma you have in early life or based on how nurturing your parents are or your caregivers or whoever. But in this case, we're talking about rats, and as a very attentive rat parent, you're highly nurtured, and you could see our nucleosomes, our histones wrapped around that DNA, are unpacking themselves. So they unraveled. They were really tightly coiled at first, but then they unraveled, and that allowed this gene to be expressed. And this is how it's going to be for the rest of its life. So this gene will be part of that euchromatin, which is that less tightly packed region. So we're going to more easily be able to express this. Okay, so this is one way that environment can directly control which genes are being expressed versus not being expressed, which is kind of crazy. So let's be an inattentive rat parent, and we're going to go, but we're not going to lick our rat pups at all. Bad parents.
Choose among the options on the left to learn more about how these mothers come from a long. Immediately after birth, nurturing affects more than the GR gene.
I goofed it up. Give me a second.
Deep in the brain.
Okay, so we're not going to lick the rat pup at all. Okay, so bad rat parent, ignoring our pups.
It looks like you had better things to do. Since you didn't lick and groom your pup very much, its GR gene is still inactive. Your pup will have a hard time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So, because in early life, this rat didn't have a parent that nurtured it and was attentive to it, that means that it's going to have tightly coiled DNA that's not going to express this protein that helps the rat relax after stress for the rest of its life. So, again, this shows that environment can have a more of an impact on epigenetic profiles than we originally thought. And this is shown in humans as well, especially in regard to the stress response to traumas earlier in life. Okay, so let's go back to this. And that brings us to the next thing, DNA methylation. Okay, so this is the next level of control. First level of control is how tightly packed we have our DNA. Second is DNA methylation. So methylation of DNA, it blocks those transcription factors, okay, which gets no transcription, but like, what does the DNA methylation mean? So basically, you take a cytosine, and it happens more frequently in our pyrimidines, which are these cytosine and thymine, especially cytosine. Just the chemical structure of cytosine makes DNA methylation really easy. So all it means is you have the normal cytosine. You're just changing one part of the cytosine, and that's instead of this hydrogen here, you're going to insert a methyl group. Okay, so that's where the DNA methylation comes from. Okay, so then it becomes 5-methylcytosine. So that methylation of DNA, just that small change in DNA, can actually impact if transcription factors are going to bind there. And think if you have a lot of C's in a sequence especially, that's going to really, it becomes more hydrophobic. For example, you're inserting more hydrogens, more carbons, and so that's going to block some of those transcription factors due to that chemical change. Okay, so this leads to it says nearly permanent inactivation of genes. There's research that shows that you can demethylate but that's less likely to occur than DNA methylation itself. And this is, it has an example here, which is inactivated mammalian X chromosome, which is a Barr body. We're going to get to that in genetics, but I just want you to keep that in mind. We're going to get to that in the next unit. I'm going to put that off for now. But you've actually, research has shown that in your cells, DNA methylation can occur if you have exposure to tobacco products. So smoking can actually make some regions of your DNA more methylated, as well as a poor diet can change the methylation of your DNA. A lot of stuff in the environment that you wouldn't normally think of as things that impact your DNA directly, they actually do. So even though it's not changing the sequences of your ATs, Gs, and Cs, it's changing the chemical of the C such that you're changing the transcription level of that gene entirely. So here's my soapbox about living a healthy life. So make sure you're exercising, good nutrition, good sleep. Sleep deprivation can lead to this as well as toxins in the environment, and tobacco being one of them, but alcohol use is also one. So being careful about uses of these substances, because it has more effects than you'd originally think, and more effects that the scientific community is learning more and more about. So it's something to keep in mind as you become adults in life. Okay, so that's DNA methylation, and here's another one that's kind of on the DNA level, which is histone acetylation. Okay, histone acetylation is the attachment of acetyl groups. You don't have to memorize what that is, but that is what the acetyl group is. It's a carbon, oxygen, carbon attached to 3 hydrogens. Okay, so what this does, all you have to know about this is that it attaches to these histone proteins. Okay, so these balls that the DNA is wrapped around, you have these acetyl groups that are attached to those. And then what happens is, because you've attached this molecule, it actually makes it more loosely unwind. And so we talk about condensing the DNA can actually make that gene unable to be transcribed. But if you have acetylation of histones, that helps unwind the DNA, and this kind of does the opposite effect. This makes sure that gene is read and unwound. So acetylation more loosely wraps around the histones, which helps transcription occur in those genes, which could be a bad thing or a good thing. It's not bad that you have some genes that are condensed, okay? Some genes are necessary to be condensed so that you don't waste energy using them in cells that don't need them. And this isn't necessarily a good thing. You could have genes that you don't necessarily want to transcribe, and histone acetylation makes those possible. So this does turn on the gene, but I don't want you to think of genes being turned on as always a positive thing and genes being turned off as a negative thing, because it's the control of those genes that is the important thing. Okay, so we're changing. Point of histone acetylation is you're adding this acetyl compound, which is making this chromatin more active, more transcriptionally active, rather than this condensed chromatin. So we're reversing the condensed chromatin. All right, so moving on to the next level. So that was our DNA level and our DNA coiling level. This is transcription initiation, and this is where we are controlling on the mRNA level itself. So we are at the promoter, okay? At the promoter, remember what happens at the promoter level. I almost asked a question as if any of you are going to respond. So I'll respond to myself. What binds the promoter, Ms. Miners? Well, it's so awkward. It's all RNA polymerase, okay? So at the promoter, you have your RNA polymerase and transcription factors. And then you have what we haven't talked about yet. We already know about promoters. We know what they do. But we haven't talked about enhancers. And I'm going to go back to that transcription video we talked about a chapter ago to talk about this more, so you can see it and how it works. But enhancers are distant control levels from the DNA, from the gene you're actually trying to transcribe. And this is where we bind activator proteins. We talked about that a little bit in your POGL yesterday. Those activator proteins are activating that gene, so it's turning on the genes. And some genes absolutely need those activators to occur. Some genes don't need activators, but others do. So you have an enhanced rate, a higher level of transcription if you have those activators present. So just because you have activators and on those enhancers means that you can transcribe more frequently, okay? So without them, you can still transcribe, it's just more slowly. Okay, so let's take a look at, I'm gonna bring up this video that we talked about before. I'm gonna turn the sound off on it. Or I don't know if I can do that, so here we go.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
So remember, this is our RNA polymerase, and
a gene is simply a length of DNA
our promoter where those
enzymes
enzymes are attaching. Our RNA polymerase is about to come in here. There it is. So we have our transcription factors and RNA polymerase binding around this DNA. So it's about to express this gene. But I want you to pay attention here, because here is where we're going to see our enhancer region and our activator region. Okay, see how, watch this in the background. This is a sequence upstream from the gene, so pretty far up from the actual gene we're transcribing. And it's going to bend back and activate that gene, okay? So it's going to enhance the level of that gene. So watch this being done.
Everything is ready to roll. 3, 2, 1.
So that right here, we have, you can see it folding up on itself. Right here, we have the activator protein binding to that enhancer region. So just to reiterate, the enhancer region is on the DNA. So it's a sequence on the DNA. The activator protein binds to the enhancer region, okay? So the enhancer region is that sequence. It's far up from the gene that we're talking about, but it bends back to make sure transcription occurs. And the activator proteins are the ones actually attaching to RNA polymerase and activating that gene. So I'm just going to go back so you can see it one more time, and here we go.
Everything is ready to roll. 3, 2, 1, go.
And then it starts transcription. So it's still the same video we looked at before.
The blue molecule.
So we're not going to watch a whole thing, but then the transcription of the gene occurred there, okay? So that's our transcription initiation. We can control how much a gene is transcribed if there's an enhancer region with activator proteins bound to it. And so here's kind of how this shows in a more picture version. So you saw in the video, and here's the picture. So you have the gene of interest, the gene we're actually talking about. You have your TATA box, the beginning of the promoter. You have your promoter. This is where RNA polymerase is going to bind, and your transcription factors, okay? So you have your transcription factors binding here. They're coming in from here. They're binding here. RNA polymerase can bind because it has those transcription factors available. But it's these activator proteins that are bound to this enhancer region that comes back and actually initiates the transcription event. Okay. So you have, that's our enhancer DNA sequences, our activator proteins, but we also haven't talked about you can either activate or you can silence this way too. So you can also, instead of having activator proteins bind to this enhancer sequence, you can have what's shown here, silencer proteins, and they also can bind to this sequence, the enhancer sequence. And that blocks transcription. So it blocks that transcription from occurring at all. So this is one way you can turn it off or on, just depending on if you get an activator or a silencer. And that's just a bigger version of it. You can see it calls them enhancer sites, but there's more than one activator protein. Sometimes there's more than one, sometimes there's just one. It just depends on the gene you're talking about. Don't worry too much about these. It just shows a real image of it. And that's a video we just watched. Okay, so now we're on step 3, which is, that's our transcription level for eukaryotes. Also, one thing that I should mention is that in, these slides are back from 2008 when Ms. Foglia was still teaching, and she has great slides, but there have been more discoveries since then. So we used to think that operons that we're talking about in prokaryotes, like the lac operon, trip operon, we used to think those only occurred in bacteria, and that's what our slides are showing. But in reality, there's actually, there's actually in eukaryotes some operons too. So that's a new discovery, is that we have similar operons to prokaryotes. We don't have specifically the lac operon and trip operon that we know of, but we have similar operons that work in similar ways. So that's another one that's not on here, but I just wanted you guys to know about it. Okay, post-transcriptional control is after transcription. So think when we know we have transcription, we have our mRNA, and we had to add the 5' cap, the 3' poly A tail, and we had to splice out those introns, okay? So this is what's being shown here on the left side. So you have your mRNA sequence. We've added the red, just shows the cap and the tail in the diagram. And then we spliced out all these orange parts, which are introns. Remember, introns in between exons are expressed. So you have all your green, and now here is your modified, fully mature mRNA molecule, which has those exons. Okay, so you have DNA. Again, this is just showing a different version. So you have all these exons put together over here. Over here, this shows two different ways that you can actually splice your mRNA. And so this is one way you can control what the resulting protein is. So you can have, if you're just talking about, they're just showing this by two different colored exons here. Okay, one is green and one is orange. So in this case, you can actually splice out one of these exons and get just this version of your protein, which only has the green exon. Or you can get a different form of splicing, which is when you just have the orange one. It's the green one that's left out. Okay, so you can also kind of switch the order of the exons. It's called exon shuffling, and that can get you an even different protein. So after transcription, you can see that occurring. And that's one way you can control what the resulting protein is and how long it's going to last. Okay, so there's exon shuffling and different ways of splicing. And a lot of those are different RNAs we talked about, where there's a lot of different kinds of RNA. Some of these RNAs are the ones that splice for you and determine which way they are spliced. So it's more complicated than we originally talked about, and we're just finding out more and more complications, which is why we are able to be complex organisms. Okay, so another thing with mRNA is regulation of how quickly your mRNA is degraded, and we already talked about this a little bit. The lifespan of the mRNA in the cytoplasm determines how much protein synthesis that can occur. So if I have my one mRNA molecule and we add that 5' cap, we add the poly A tail, we're splicing out the introns, okay? So here's our final mRNA that's going to go through the ribosome and be made into a protein. Well, say this poly A tail and this 5' cap are pretty long, especially the poly A tail is what matters here. Say this poly A tail is really long, it's going to last longer in the cytoplasm, and it can go through ribosomes more than once. So this protein can be expressed multiple times. Okay, this mRNA can be expressed multiple times, excuse me. If you have a short poly A tail, however, then you're going to have those exonucleases we talked about eating away at the ends of that. So it's not going to be able to go through the ribosomes as frequently, as many times, so that controls how much of that protein is being expressed. How many times that mRNA is able to go back to the ribosome and make the protein. Remember, those exonucleases are the guys that are kind of eating the ends of that. All right, next is RNA interference, and this has to do with these guys are targeting mRNA. Okay, this is one type of RNA we're going to talk about, which is siRNA, or small interfering RNAs. And these are really short segments of RNA. And what these guys do is they actually bind to mRNA, and they create, so here's our siRNAs, little snippets, just little guys, okay? But they can bind, here's our mRNA molecule, these long snake-like green guy. These siRNAs can actually bind to the mRNA and kind of based on the sequence that's there. So they're going to base pair with whatever sequence is matching, is complementary to them. And what that does is it makes it a death tag. So you can kind of think of these siRNAs as they aren't doing the, I don't know how to say this. They are not doing the killing of the mRNA, but they are tagging them for death. So think of like a gangster, okay, mob boss. Mob boss is the guy that makes the call that says, hey, I need you to take this guy out, okay? Well, the siRNAs are basically the call that's made to take the guy out, but they're not really the ones taking him out. The ones taking them out, therefore silencing the gene by degrading the mRNA, is this guy, which is our breakdown enzyme. So the enzyme, think of this enzyme as the assassin that's hired to take out this mRNA, okay? So the siRNA is the one this is the call that says, hey, I need you to take this guy out. So it finds where that mRNA molecule is. It says, hey, this is our guy, this mRNA. And then you have your assassin, which is this enzyme, which is actually going to degrade the mRNA itself, okay? So by this siRNA is going to do gene silencing this way because you're basically destroying the mRNA. If you don't have the mRNA, it can't go through the ribosome and you can't make that protein. So that's yet another way. And also she says this is a hot new topic in biology, but in 2008. So I think this is more than 10 years past that. So it's still interesting, still more research being done, but it's not a hot new topic. We'll talk about some hot new topics next week. All right, controlled translation is the next one. So this is when you're actually at the ribosome. And you can block an initiation of a translation stage. So you can have a regulatory protein actually attaching to the 5' end. And if you're blocking that 5' end, you're preventing the attachment of the ribosome. And that initiator tRNA, which is going to put that methionine on there. If this guy can't start the chain, then you won't have that mRNA going through the ribosome, and you won't have that protein being made at all. So you're just blocking translation. This one's one of the more simple ones. Okay? And these are our last ones, okay? This is where you can control at the protein level. And this being said, this is probably you're wasting more energy this way because the longer you take to actually regulate, the more energy you're using, the more ATP you're using in the cell to make this protein when you didn't actually need to. So the earlier it's regulated, the more energy is saved by the cell. And once again, that's why bacteria actually regulate at the transcription level, so they don't have to do this. It can serve some energy. Okay, so 6 through 7, these are our last ones. This is protein processing. So you're actually regulating at the protein folding level and how the protein folds and how quickly the protein is degraded. Okay, so you can actually add sugar groups, this is what it says right here, adding sugar groups, and by whatever compounds you're adding to that protein, you're affecting how that protein is folding, and you can deactivate that protein entirely, or it can be turned into something else. So that's another form of regulating is if it doesn't fold right, then you're not going to get that protein. Okay, so if you're changing the chemical structure of the protein, you're changing how it's folding. And that's kind of what we were doing. I know we were rushed that day, but we had those pipe cleaners showing how it was folding, and you can get different forms. You can get those folding in different ways, and it all depends on that chemical structure. And then you have protein degradation, and this is ubiquitin tagging. This is kind of like, I'm going to use just the same example. You can use a different analogy if you want. But the ubiquitin tagging is kind of like the siRNA for mRNA, except this is for a protein. So the ubiquitin can actually tag a protein. And if ubiquitin, which is the mob boss that makes the call, says, got to take this guy out. Protein is the guy being taken out this time. Ubiquitin tags onto this protein, then it's going to be chopped up by a proteasome. Okay, so this is the assassin this time, is this proteasome. And it knows to basically eat that protein and chop it up if it's got this ubiquitin tag on it. So no ubiquitin tag, good to go. Ubiquitin tag, done. Proteasome destroys it. So this is just an enzyme that's in the cytoplasm of the cell, destroying proteins that are unneeded. And this is another way that you can regulate. And this is a death tag, just sounds like really, really scary. So this tells you about the guys that actually discovered it. And this is kind of what it looks like. It looks kind of like a tube. And you can think of it like a garbage disposal, okay? You're done with it. You don't want, generally the cell doesn't want that protein anymore. It's done serving its function, or it's no longer helpful for that cell to have that protein doing its thing. It could be an enzyme. It could be initiating a stress response that you don't want to anymore. Like adrenaline protein, epinephrine, you don't want to have that initiating processes in the cell. You're done with it. So you might want to tag that with ubiquitin and destroy it by the proteasome so you don't have that stress response happening all the time. Okay? So this guy's a garbage disposal that basically goes, all right, we're just going to take this and chop it up. And then we're going to, again, remember, you can reuse these amino acids. Okay? So it is kind of like cellular recycling, as it says here. Because you chop it up. Oh, let's reuse these amino acids for things that we actually need. Okay, so that was all our levels of, thank you, I will get mushrooms at the store. That's all our levels of gene regulation. So just to review really quick for you, we have our coiling of the DNA. That was our step 1, how much it coils around those histone proteins, how condensed the DNA is, affects if transcription can occur. Step 2, you have the actual initiation of transcription, so whether that gene is transcribed or not. 3, you have alternative mRNA splicing, so you can have exon shuffling, you can have different exons in the mRNA being spliced out. So different splicings, different regions of splicing will change the protein that you get. You can have mRNA protection, so that's the poly A tail and the kappa, mostly the poly A tail. How long it is depends on how much it can survive in the cell. So that determines how many times it can be made into a protein. You have mRNA processing and initiation of translation, so you have the blocking at the ribosomes. And then you have protein processing, so affecting the folding of the final protein, and degradation, which is finally sending it into ubiquitins so it can be chopped up. And that's all. I'm going to have you, we're going to do more on this stuff. We're going to do some activities with it, but I'm not going to go over this in class. So remember, if you want to rewatch this, it's always going to be on Google Classroom, but I'm not going to go over all of these things again in class. So there you go. Hope you enjoyed this rendition brought to you by, again, the Washington Coffee Shop, which my coffee is now a little cold, so I'm a little upset. Maybe the next video lecture will be brought to you by Mr. Miners. Who knows? Enjoy your snow day. Maybe we'll be off tomorrow, maybe we won't.